Hello, Mindy again. So we continue the series on the histology of the male reproductive tract, and we see we have a transition around the prostatic urethra, and this transition zone is mainly prone to benign prostatic hyperplasia. Then we have a central zone that is around the openings of the ejaculatory ducts, and this comprises 20% of the gland. The transitional is only 5%, contains only 5% of glandular tissue. Then the bulk of the prostate gland is mainly the peripheral zone at the periphery, and this region is prostatic cancer. The muscular portion contains no glandular tissue. So it's still center to divide the gland into lobules. What are the functions of the secretions of the prostate? It contains bicarbonate and proteolytic enzymes, citric acid, zinc, lipids, and a variety of proteins. We also have clotting enzymes that act on semen to form coagulum. We have profibrinolysin that liquefies the coagulum. We have prostatic acid phosphatase, which is a tissue-specific marker. We have prostate-specific antigen, which is a proteolytic enzyme that liquefies the coagulated semen. So after the semen has coagulated, we have prostate-specific antigen, a proteolytic enzyme that is able to liquefy the coagulated semen. The prostatic fluid helps to neutralize the acid vaginal secretions in the female tract. So the structure and function of the prostate are also dependent on dihydrotestosterone, which is an androgen. We go to the bulbourethral glands of Cowper. These are small glands with ducts that enter the membranous urethra. Remember, we have 4 parts of urethra: prostatic, 3 parts prostatic, then membranous, then the penile urethra. So these bulbourethral glands enter the membranous urethra. They secrete clear, viscous, and mucus-like, rich in sialoproteins, and they serve as lubricants for the urethra. So bulbourethral gland secretions serve as lubricants for the urethra. They are the analogues of the greater vestibular glands of Bartholin's in females. So the Bartholin's in females are homologues of bulbourethral glands of Cowper. The structure and function of these glands again depend on androgen. We go to the penis. This is a copulatory organ. There are 3 cylinders of erectile tissue in the penis, as well as the penile urethra. The cylinders are usually enclosed by connective tissue and skin. These 3 erectile tissue cylinders are enclosed by connective tissue and skin. So we have a single corpus spongiosum. This usually encloses the urethra and enlarges terminally into the glans penis. So corpus spongiosum is the first cylindrical erectile tissue. So single corpus spongiosum is just one. It encloses the penile urethra, and at the terminal portion, it enlarges to form the glans penis. Then the other 2 erectile tissues are the paired, parallel, and dorsal cylinders of erectile tissue, which we call the corpora cavernosa. So that's why we have 3, 2 corpora cavernosa and single corpus spongiosum. So you have 2 corpora cavernosum and a single corpus. This corpus spongiosum contains the urethra, and you can see the vessels within them, the bulbocavernous artery in the corpus spongiosum, and the corpus cavernosum has the cavernous arteries in them. And then on the dorsum of the penis, you have the deep dorsal artery and the veins. Okay, the artery and the veins, and then you also have a superficial artery. So there's skin followed by superficial fascia, areolar tissue before you get to the deep fascia and the erectile tissue. So the corpora cavernosa are 2 cylinders of erectile tissue. The caudal ends form the crura of the penis, while the rostral ends stop at the glans penis. Each of these corpora cavernosa is enclosed by thick, dense fibroelastic tunica virginea. Each corpora cavernosa is enclosed by thick, dense fibroelastic tunica virginea, and partition between the 2 cylinders is incomplete. The partition between the 2 corpora cavernosa is incomplete. Therefore, there is communication between the vascular spaces. So the erectile tissue has a vast honeycomb-like system, which we call the cavernous blood spaces. these are erectile tissue. Corpora cavernosa are made up of erectile tissue. But what do we mean by erectile tissue? It's a tissue with honeycomb-like system or irregular blood spaces, cavernous blood spaces. And the walls of these spaces are what you call trabeculi. They are made up of smooth muscles, nerves, and dense connective tissue. So these vascular spaces within the erectile tissue, they have walls called trabeculi. So these vascular spaces have walls called trabeculi, which are made up of smooth muscles, nerves, and dense connective tissue. And since they're blood containing channels, they're lined by endothelial cells. Now, we have deep arteries that are running at the center of each of the corpora cavernosa. And we also have major veins at the periphery beneath the tunica albuginea. So this erectile tissue is similar to the pleura of the clitoris of the females. Now, the corpus spongiosum is just a single cylinder of erectile tissue. It's blunt at both ends. The rostral portion enlarges to form the glans penis. The caudal portion forms the bulbous penis. Now, there's a thick tunica albuginea that is surrounding the corpus spongiosum. Then we also have trabeculi with more elastic tissue and less smooth muscle. So remember, the trabeculi, the walls of the vascular channels within the erectile tissue. So in the corpus spongiosum, they are more elastic and have less smooth muscle cell. Now, in the center of this corpus spongiosum is the penile urethra. So this is histologically, this is how the penis looks like. Definitely this is corpus spongiosum because of the presence of urethra. How do you know this is urethra? The transitional epithelium. Then the corpus cavernosum, two of them. Okay, you can appreciate the vascular endothelial lined channels. Okay, and their walls are the trabecula that have smooth muscle, nerves, and connective tissue. But we call this, all these are erectile tissue because of the presence of these vascular channels, which when they are filled with blood, they cause erection. So erectile bodies, all these are vascular channels filled with, when they are filled with blood, they cause the penis to erect. That's why we call them erectile bodies. So what is the mechanism of erection? Now you need to remember that we have two types of blood arteries in the penis. We have nutritive vessels, of course, nutritive, so they will feed the tissues with oxygen and nutrients, but we also have helicine arteries, which are afferent. So these helicine arteries are the ones involved in erection. So helicine arteries bring blood and empty directly into the cavernous blood spaces. So what happens during erection? The cavernous blood spaces become filled with blood. So when there is sexual stimulation, these cavernous blood spaces are lined by trabecula. So when there's sexual stimulation, these trabecula relax. Then helicine arteries now push blood into these cavernous spaces. These spaces will be filled up with blood and hence making the erectile tissue cylinders to be more rigid. The outflow of blood from the cavernous spaces via venous drainage, which is now efferent, is usually retarded by the resistant tunica albuginea. So during erection, there is no venous drainage, or there's reduced venous drainage. Why? Because the tunica albuginea is now is resistant, so it retards venous drainage. Therefore, erection is maintained when there is greater rate of blood flow than outflow. So when the cylindrical erectile bodies become rigid because they are filled with blood, when they become rigid, they impede venous drainage because even the tunica albuginea is resistant. So inflow becomes more than outflow, therefore erection of the penis is maintained. Now, after ejaculation, after sex climax ejaculation is achieved, that means there's the removal of stimulus. So the smooth muscle cells of these trabeculi, they now tense. Remember, initially in erection, they were relaxed to allow more blood. They now tense, they contract up. These smooth muscle cells tense up, contract. And when they contract, they now pump blood out. So outflow of blood exceeds the inflow and the penis becomes flaccid. So in a flaccid penis, the smooth muscle cells of the helicine arteries and smooth muscle cells of the trabeculi, their tonicity is increased. So you're able to keep the blood out of the cavernous space. So corpus spongiosum does not reach the same degree of rigidity as the corpora cavernosa during erection. So the return of the penis back to the flaccid state is what we now call detumescence. We have two chemicals that are responsible for detumescence, nitric oxide and phosphodiesterase. Nitric oxide is produced by the nerves in the penis. It will cause relaxation of smooth muscle cells. Okay, and these smooth muscle cells, when they relax, erection occurs through the production of cyclic GMP. Now, we have phosphodiesterase enzyme, and this comes and degrades cyclic GMP. And when you degrade this cyclic GMP, there is no more relaxation of smooth muscle cells. Therefore, erection is terminated. Okay, so nitric oxide produced by the nerves of the penis, they cause relaxation of smooth muscle cells of the vascular channels in the bodies. So when the smooth muscle cells of the trabecula are relaxed, you allow in more blood. And this is through the production of cyclic GMP. So when you allow more blood, there is erection. Now, after some time, phosphodiesterase enzyme comes in. It degrades this cyclic GMP that regulates relaxation of smooth muscle. Therefore, you terminate erection. So phosphodiesterase helps detumescence to occur. Nitric oxide and cyclic GMP responsible for erection. When we go to the urethra, the first part of the urethra is a prostatic urethra passing through the prostate. It has a colliculus seminalis and a utriculus masculinus. So we have ejaculatory ducts and prostatic ducts that open in the prostatic urethra. The epithelium of prostatic urethra is transitional epithelium but becomes pseudostratified or stratified columnar at the distal portion. Then we have two layers of smooth muscles, the inner longitudinal and outer circumferential in the prostatic urethra. The outer layer is highly developed at the internal urethral orifice, and this forms the internal sphincter of the urinary bladder. So this is your prostatic urethra. You can appreciate the prostatic utricle and the two openings of the ejaculatory ducts. Then we have openings of ducts of the prostate gland itself on the side. So prostate gland openings are here, while the openings of the ejaculatory ducts are at the center of the prostatic. So the seminal colliculus contains opening of prostatic utricle and ejaculatory duct. The gland itself opens at this area. So this is the prostatic urethra within the prostate, prostatic urethra, then membranous urethra, followed by the penile urethra. Again, you have your prostatic urethra, then membranous urethra at the urogenital diaphragm, and it's the membranous urethra where you are able to appreciate bulbourethral glands, and then that's the penile urethra. So the membranous urethra is very short, lined by stratified columnar epithelium. Smooth muscle layer is thinner, and the striated muscle fiber of the urogenital diaphragm surrounds membranous urethra, and this forms the external sphincter of the urinary bladder. It comprises the sphincter muscle of the urethra, forming external sphincter of the urinary bladder. Then the penile urethra or cavernous urethra, it's the longest portion of the urethra. It expands at the bulb of corpus spongiosum, which is the bulb of the urethra, and in the glans penis. So it has two enlargements at the bulb of corpus spongiosum and at the glans penis, forming the terminal fossa. The epithelium of penile urethra is stratified columnar, and at the distal portion, the fossa outwards becomes stratified squamous, non-keratinized, and in turn becomes continuous with the stratified squamous keratinized that covers the gland penis. So epithelium is initially stratified columnar, then becomes stratified squamous, non-keratinized. Then as you get outwards at the glans penis, it now becomes stratified squamous keratinized. We have Litters glands, which are small glands containing mucus secretory cells that open up in the penile urethra to aid in lubrication of the urethra. So the semen is a complex fluid from the testes and accessory glands. It's thin and milky emulsion, and the testes and the ducts contribute to the fluid with a high concentration of sperms. The seminal vesicles contribute a fluid that is high in fructose and prostaglandins. So semen contains secretions of prostate, seminal